Hello, everyone, and welcome to the first Biosat chat of the recurring series Scientific Collaborations Across Borders, organized by QBI and the Office for Science and Technology at the French Consulate in San Francisco. So thank you very much for joining us today. I'm Héloïse Pajot, Deputy Attaché for Science and Technology at the French Consulate in San Francisco. Thank you very much for joining us today. So I'm very glad to have Neven Krogan, Director of QBI, and Marco Vignuzzi, Principal Investigator at the Department of Virology of Institut Pasteur in France. So today we're going to discuss the importance of scientific cooperation across borders, the challenges, but also the solution for conducting successful international collaboration. And for that, what a better example than the collaboration between Institut Pasteur and the QBI Coronavirus Research Group. Before diving deeper into this discussion, we have the pleasure to welcome with us today Frédéric Jung, Consul General of France in San Francisco, to launch this first session. Frédéric, the floor is yours.
Thank you so much, Héloïse, for this introduction. Good evening for those of you who are joining us from France, and good morning to those who are in San Francisco. Thank you for joining us today. I'm Frédéric Jung. I'm the Consul General of France here in San Francisco, and I'm delighted to be here with you to launch the first session of what you said, Héloïse, the Scientific Collaborations Across Borders Fireside Chat series. This series is co-organized by QBI, the Quantitative Biosciences Institute of UCSF, and the Office of Science and Technology of the French Consulate General in San Francisco, in a common goal, which is to highlight scientific collaborations between France and the US. These sessions will be held every 6 weeks throughout 2021 to share experiences, discuss challenges that have to be overcome, but also the solutions that have been developed to carry out successful scientific collaborations. These discussions are a great opportunity to share and highlight what is possible when we gather our strengths for the same goal. The collaboration between Institut Pasteur and the QBI of UCSF coronavirus and the QBI Coronavirus Research Group during COVID-19 is a true example of successful collaboration across borders. Indeed, in 2020, as we were in the midst of a pandemic, UCSF's QBI director, Neven Krogan, joined forces with researchers from UCSF, from Institute at UCSF actually, also with the Institut Pasteur, with the Icahn School of Medicine of Mount Sinai in New York, with the Howard Hughes Medical Institute, and they applied their expertise to aid in finding a treatment for the growing pandemic. And together with this group of researchers, which is now known as the QBI Coronavirus Research Group, they were the first to actually extensively map out the genome of COVID-19 and discover that the virus actually interacts with 332 human host proteins. And the team accomplished this in a matter of weeks only, which usually takes years. So thank you, Neven and Marco, for your work, and also thank you for being here today to share your experience. To conclude, I hope that the series will give useful insights to strengthen our current scientific collaboration, but also foster new ones between France and the US. I now give the floor back to Héloïse. Thank you again, and I wish you a very good session. Thank you very much.
Thank you very much, Frédéric, for these welcoming words. So before we start, I want to welcome our viewers who are joining us live to not hesitate to ask questions at any time using the comment section on Facebook or YouTube. And I will be happy to relay your questions to our panelists and get you involved in the discussion. Okay, so to get us started, Neven, Marco, could you please each briefly introduce yourself and tell us a bit more about who you are and what you're doing? Neven, would you like to start?
Sure. I'm very happy to be here. Thanks, Héloïse. So I am a professor and director of the Quantitative Biosciences Institute here at the University of California in San Francisco. I'm also an investigator at the Gladstone Institutes here, which is affiliated with UCSF. And a big focus of the research that ongoes in my lab and also in the Institute is the development of technology that allows for the quantitative analysis of molecular information of various kinds. And it's a very disease agnostic type of approach that can and is being applied to many different disease areas, including cancer, neuropsychiatric disorders, heart disease, and infectious disease, which is obviously the topic of today's conversation. So we're in a great position to pivot rapidly with our disease-agnostic technology in collaboration with many groups around the world, including the Institut Pasteur, to study SARS-CoV-2 to get a better understanding of it and to point us in important novel therapeutic directions.
Hi, and I am Marco Vignuzzi. I am a principal investigator, a virologist within the Department of Virology at Pasteur Institute. And my lab primarily studies RNA viruses, not particularly coronavirus until the pandemic, but we cover a wide range of viruses of human interest. And much like most of the virology world, with the arrival of the pandemic, we were thrown into studying this new virus and had relatively little time to ramp up as much as possible, really dedicating 24 hours a day, 7 days a week for the last 1.5 years to work on this virus. So in this discussion, I think I'll be able to represent much of the world of virology and the challenges that we encountered in trying to quickly adapt our knowledge to a new virus and then move from there in any direction possible. In my own case, my lab generally studies virus evolution. And in this particular year, we focused on other things. I'm really working with Nevin on protein interactions.
Yeah, thank you for this introduction. So you both collaborated about COVID. Could you please tell us more about this collaboration and the QBI Coronavirus Research Group?
Sure, I could start here. I should probably point out that this connection that's been so fruitful during the pandemic with Marco and his colleagues really started several years before the pandemic. At QBI, one of the goals is really, if not the major goal, is collaboration, is making connections between different scientists here on campus, across the United States, and really around the world. And a couple of years ago, we started with Olivia Schwartz, who was the scientific director of the Pasteur at the time, and now it's gone on to connect with Christophe Danfer, who's now the current scientific director at the Pasteur. We set up a collaboration between QBI and the Pasteur Institute with the real goal is to try to bring together the different expertise, the strong infectious disease expertise at the Pasteur and a lot of the technologies that we've been developing here in California. And along those lines, we had a symposium here in San Francisco where a number of scientists came over from the Pasteur. That was back in March 2019. Marco wasn't one of the ones that came. He was going to come next time. And we were actually going to have another symposium in Paris, and then the pandemic hit, and we had to do that then virtually. But we had kind of set this up. We had set the stage in many ways over the last few years. And then when the pandemic hit, we were in a perfect position, I would argue, based on our collaborative expertise to work together in a very quick and facile way. And I should also point out, as Frederick alluded to, it wasn't just with the Pasteur. We also connected with other scientists in New York, in England, in Scotland, and many other places around the world as well. So a lot of the groundwork was set in the previous years, and that's why one of the major reasons why we could respond so quickly.
If I can jump in and speak a little bit of Pasteur's side, there are two primary things that prepared us for this interaction and this pandemic. The first is, of course, Pasteur Institute is over 100 years old. It was founded by Pasteur himself in the late 1800s, and it's always been an institute specifically centered around infectious disease of humans, whether it be bacteria, parasites, viruses, etc. So we have 120 or so labs or PIs that work in essentially this field. So of course, when the pandemic arrived, even within Pasteur itself, the Department of Virology has over 20 teams working on various viruses of human interest, including the previous coronaviruses, SARS and MERS, but also on campus, there's a whole other group of people, public health researchers, epidemiologists, modelers, immunologists, all kind of centered on these questions. So the ability to work on this virus was there, but we didn't necessarily have the strengths that QBI had within its own walls. The second thing about Pasteur is that we're very, very much already in tune with the globalization of research and international interactions. Pasteur Institute, although many of you may know the Paris campus, we are actually an international network of over 30 institutes on all of the continents in the world. And so we have daily interactions with all of these institutes that really, again, encompasses the main goal of tackling issues of public health, and particularly infectious disease. So in the last few years, as Nevin mentioned, there was interest in finding a way to bring Pasteur Institute and QBI closer together. The pandemic ultimately offered a springboard to the original ideas that were being put together by Nevin and the scientific directors at Pasteur.
And maybe just along those same lines, I'll add just a little bit more description of QBI and how it's synergized with the Pasteur. There are over 30 labs in the QBI building and another 70 labs affiliated. So there's about 100 laboratories connected to QBI, and over 40 of them, when the pandemic broke, decided to come together and bring their diverse expertise to the table and in a very integrative ways to study this virus and hopefully come up with solutions on how to fight it. And there's a picture of the PIs that has just come up. And so there's hundreds of different scientists. We actually broke this into a number of different subgroups. Some of the leaders here at UCSF, Kevin Choquette and Brian Choquette on the drug discovery side, and Klim Verba and Oren Rosenberg and Dave Agard on the structural biology side of things. We built up this infrastructure, and science was being done really at an unprecedented rate here in the Bay Area. A lot of the motivation was out of fear. And then we leveraged these connections. And one major one was obviously with the Pasteur Institute. We didn't have the virus growing here for some time. We do now with collaborators like Melanie Ott at the Gladstone, but at the time we just didn't have it. But the Pasteur Institute obviously is one of the best virology institutes in the world. So, pick up the phone and we say, look, we can study these proteins in certain ways, the viral proteins. We can see which human proteins the viral proteins are talking to. We can make predictions about drugs and compounds, but we weren't in a position to test those and do kind of the really deep virological assays that were required. So then we were able to, with the help of the consulate and actually the president of FedEx, to get these drugs and compounds to Marco and his team and his colleagues at the Pasteur Institute, and he's able to test these very quickly. So it was, in my mind, you can't define a better collaboration because we brought different skill sets to the table and we integrated them together in a very effective way, in a very fast and actually ultimately very fun way as well.
Yeah. Do you want to add something, Marco?
I was going to say, you mentioned getting the president of FedEx. That actually was incredibly important. Perhaps one of the challenges that one wouldn't necessarily come to think of when a pandemic is approaching, and the travel stops everywhere, and that essentially the lockdowns were making it difficult to move anything about. And it was really key that, we were having, I lost count of how many shipments you sent to us after there were over 25, each with dozens and dozens of compounds to test. That led to an astronomical amount of virology assays that were performed in my lab. When the pandemic started, on Pasteur's side, of course, we saw it in the horizon. Right at the beginning, when the first cases were being described in China, everyone had the hunch that this one is coming worldwide. So Christophe D'Enfer and the virologists and other researchers at Pasteur put together a task force, which put together sub-task forces, which really led to the whole campus working on this question. And at that time, there was, with the lockdown, of course, labs themselves were restricted, right? Not every researcher could actually run their lab. It had to be focused to only the labs that could work on coronavirus. but roughly 10% of our campus was still in the labs working. The remaining people were at home actually kind of performing the duties that these people no longer could do, so supporting each other. And when at the beginning, as you mentioned, Nevin, Pasteur was among the first to get the virus isolated and growing. This was through Sylvie van der Werf's unit and Jean-Claude Leguera. Both of them were isolating the virus and trying to deal with the diagnostics to help France ramp up its diagnostic capacity. You can imagine the requests coming from around the world, from both industry and academia, to collaborate. So what happened is we just started dispatching to the labs that were functional in Pasteur to see who could pilot the studies, and I happened to come across Nevin and very willingly joined in. And that led to a massive undertaking that could only be done thanks to the big network that Nevin put together. Even in the virology side, our lab did everything we could to test every single compound, but it was helpful in having at Icahn School of Medicine, Mount Sinai, Adolfo Garcia Sastra's lab doing the same thing in parallel.
Yes. So thanks to this big network, you were able to have different expertise from all around the world, and this was very useful. But I can imagine that collaborating with a different country can be challenging. Like, for example, maybe you have the barrier of the language, the difference in the culture. Maybe you don't work the same way. So could you please tell us more about this experience on collaborating across borders? And perhaps if you have maybe good practice to share with us to foster more international collaborations.
Sure, I could start. I guess, the biggest barrier is the time difference. But since I think Marco and I haven't really slept that much the last year and a half, I don't think that was such a barrier. But one of the great things, obviously, the pandemic hitting now versus maybe 30, 40 years ago is the technology and the way one can communicate so effectively through different platforms like the one we're on right now. So to me, it was actually a lot easier than I guess I would have anticipated. Obviously, we've been collaborating in the past. We collaborate with people around the world, but never at this speed and never to this extent. But to me, it was actually a fairly easy. I mean, English is the one language that's prevalent throughout science. So obviously, we were all speaking English. But to me, it was actually amazing to see how well we could communicate with mechanisms like Zoom, and I know a lot of scientists use Slack for communication and other type of platforms such as that. When there is motivation to communicate, the mechanisms were there in place. So for me, I'm kind of rejuvenated, and as we now go and focus on other disease areas and other viruses, say with Marco, we can communicate a lot better than I previously thought before the pandemic. And I think that's one of the themes in general, is what we've learned during this pandemic, we can and will apply in the future to study all diseases, essentially.
Yeah, I can add, there aren't as many barriers as one would think vis-a-vis the, everyday life. One thing is that science is international already. Scientists are an international group that have nationalities ultimately, but in our process of going through our undergraduate, master's, PhD, postdoc, and then first faculty position, second, most of us end up having to move around, not only within our country, but to other countries. And so by the time we reach my level, for instance, we've already accumulated a certain number of experiences and have kind of become globalized or international, so to speak. So the labs themselves, regardless of what country they are in, they tend to run at least somewhat in English, if not entirely in English. The language barrier isn't there. But then I suppose you can, if you really, one were to study it, you would find dialects, let's say. And I'm not talking about the language, but more the lab culture. It's true that how one approaches science and scientific questions can be influenced by the type of education system that you come from. And then you accumulate other experiences. That won't necessarily be obvious in something like the work we did during the pandemic, where really it was go as fast as you can, and you don't really have time to put your own little spices onto the research. But in my own case, for instance, I did my upbringing and growing in Canada. I moved to France and did all of my graduate studies in France, did a postdoc in the United States, and then came back to France. And I did see differences in how the education systems teach you to approach a problem. If anything, we benefit from going through two different systems where you have to kind of forget what you learned and relearn a new way, go through the experience of feeling dumb and then smart again, that kind of thing. But ultimately, it does help you round out your approach to science. Now, in the specific case of this pandemic, really it was, you had a job to do and it was very clear. Unlike in regular circumstances where maybe my lab would attempt to do all of the work and therefore we'd have to learn how to do some of the techniques we didn't know, that would take a long time. Here it was like, just let every expert do their thing, not try to be the king of it all, and that was clear from the get-go. So in that sense, there was really just kind of the outline of what we had to do and just the bullet list of what had to be done.
Yeah, and I guess it's, as you say, Marco, you did your postdoctoral fellowship here in California. So you were familiar with the California dialect of science, and you were familiar with a lot of the characters that were connected to QCRG because you were here. And I think that helped out a lot. Plus you were in Canada, and I'm Canadian too, so that was good. That was a good connection. And then also just the different styles, I think it's worth pointing out, of science between, let's say, the United States or California and Paris. And I think I would say there's pluses and minuses to both styles. And it makes it very complementary, in my opinion. I would say maybe, I'd like to hear Marco's comments on this too, but France is a little bit, it's careful, and that's good. That can be very good, and it's methodical. But I think, as you said, Marco, when I was talking to you yesterday, the American styles, maybe you take more of a leap of faith. You can a little bit more brute force, and I think, as I said, there's pluses and minuses to both sides, and you put them together, these two different styles. There's a lot that can be accomplished, and that was evidenced from all the great work that QBI and the Pasteur Institute did over this pandemic. I don't know, Marco, did you want to add anything to that?
Well, I can really give my two experiences, just little anecdotes. Throughout my whole undergraduate studies, where I came fresh to the French system that really gets you to fit into the box and do things the right way, I was messy, and I just remember, I can't count the number of times Sylvie van der Werf was my PhD supervisor, the number of times I had to hear the word rigoureux, rigorous, nailed it to my head, and I still get chills anytime I hear that word because I simply wasn't rigorous. And it was, it's the fact that in France, you do things very methodically, carefully, not to make the mistakes on the little steps, and be sure of what you're doing. It's almost, we do have a fear of being wrong. And so we have a difficulty admitting when we are wrong. And so this is something that, it could be a negative side, but it's also in the positive. It really makes sure that when you get to your answer, you're sure of what you're doing. You're not taking risks. On the other hand, when I came to California and I was in Rolandino's lab, it was the polar opposite of really just go for the big things and then try to fill in the spaces in between. And I remember driving with Rolandino to a conference, and he was describing to me this, this need to take a leap of faith. And kind of running to the edge of a cliff, and you have to jump, right, to get across. Of course, I'm trying to process this as a postdoc and say, yeah, but I mean, what happens if you don't reach the other side? He said, well, hopefully the pile of dead postdocs under you will break your fall. So I'm like, well, okay. Again, could be seen as a negative thing. But trying to put humor into it, ultimately, there are. It's kind of the American thing of go big or go home, to the more European style, which is maybe it has a lot of historical experience of failure, and so you want to be careful in the moves you make. The two obviously work really together, which is kind of what I try to do in my lab, and kind of marry the two. And we can only benefit even from other nations and other education systems that do yet a third or fourth approach.
And yeah, listening to Marco, just one thing to add, I think kind of another way to look at the differences is, again, in France, it's a lot more traditional. It's got this great history, and I would say it's very hypothesis driven. You come up with a theory and then you test it and you say it's right or wrong. Well, the technologies that I've been alluding to and that we have at our disposal, they're non-hypothesis, they're unbiased. You just can take these tools and systematically apply them to a question or a disease, collect a lot of information, and then use that information to come up with hypotheses. Okay, so you can see how it's complementary, but kind of different. And again, putting them together, we can learn a lot from each other, and I think that's one of the messages that we got at this pandemic is the complementarity of different approaches, different people and different cultures coming together to tackle this pandemic.
Yeah, exactly. And I think your collaboration is a good example of that, because with the QBI Coronavirus Research Group, you accomplished in a matter of weeks what was possible, what was usually taking years. So you were faster than usually. But do you think that this pace and this kind of international collaboration will be possible to continue in the future? Or is there anything that have to be changed?
Yeah, I could maybe start. I would say not just international collaboration, but collaboration even nationally here, and a lot of it comes down to funding, money, and how do you get the funding to do the research? And I would say one of the biggest problems about the academic enterprise that Marco and I and others are in is that it's very individualistic, that with respect to the reward system. One person gets a grant, one person gets an award, one person gets tenure. And in that framework, collaboration is often, I would argue, discouraged. And I would say that that's particularly pertinent for younger scientists who have these young, fresh ideas, and they're enthusiastic, and they're coming in, and then they become quite guarded because the system, as I just outlined, in my opinion, is against collaboration. So I think it would be really fantastic to flip things around. And I know it's not just an easy switch here to flip, but if the reward system could change, where collaborations could get funded instead of the individual. And I think internationally, looking at different governmental agencies, I think they could take a big push in this area by getting together and seeing the success that's been had in COVID with us and many other groups, and put funding specifically more into groups coming together with different expertises in different disciplines and in different countries. I think that would go a long way, and I hope more of that happens.
Yeah, I agree with you entirely. One of the issues, and it remains to be seen if this will change or if things will go back as usual. It'll be difficult to change things, right? So the first thing is as you said, funding recognizes the last author, and for our papers, we have at least 6, if not more.
Or more. There are many of them. They're more. They're like 12.
You can't cover everything. I know in my own experience, because after this year, where I have never had such a great publication record, but ultimately, I did apply for funding, and based on those papers, the comment was ultimately, well, we don't know what fraction of the work actually is done by Mr. Vignuzzi. So it ends up being that it's hard for people that are using the usual way of evaluating to weigh this out. So all that ultimately has to change. And another problem is that while there are opportunities for funding, they come from different sources which tend to be national, right? And so even within France, for instance, funding from the major agencies goes to groups and can pay other collaborators within France, but cannot cover international collaborators. there's other, smaller grants perhaps that are available, but ultimately there's no such thing as a world grant, where even within Europe, you can get funded within Europe, but not between North America and Europe or Asia and Europe, and so these are missing ultimately. I think the final thing that's important to consider is what is the capacity of the world to fund research in all of the other areas more than just the current pandemic. This was a pandemic where we poured all of the money, most of it redirected from other research, into it. But at the same time, there's still other diseases that were happening, right? I mean, even within virology, there were Ebola outbreaks during this pandemic that were kind of put aside because we were all focused in one thing. So the fact is that the amount of funding is still too little. I have to say, coming out of this pandemic, Europe reduced the science budgets. So, shame on us. We need to consider all of these things. At least, though, the pandemic shows that it can be done. We can drop our egos. And for the first time, I've really experienced that this was a pandemic where people dropped their immediate needs and egos and worked in total collaboration, sharing everything, posting things as quickly as possible, not worrying about competition. So it shows that we can do it.
Yeah, exactly. Thank you. I think it's a very important point to highlight. So thank you for that. We're going to go ahead with the question from the audience. We have a question to Nevan. Is there a treatment for COVID-19 better than remdesivir?
Well, obviously the vaccines are having a huge positive impact, but in terms of drugs that are available, I think remdesivir is the one, at least that's approved by the FDA here in the United States. There's dexamethasone is a steroid that's better acting later on in infection, but I know of a number of drugs that are late clinical trials that look quite promising, including one that we had worked on with Marco and Adolfo and Kayvon and Brian, and it's this one drug called plitidepsin or Aplidin. It's an anti-cancer drug from the Spanish company PharmaMar that came from our data and was tested by Marco and Adolfo in Paris and New York, respectively, to be potent in cells and then in mice. And there's a phase 2 clinical trial that's just completed. The data looks great. It was actually just posted on medRxiv, I think today or yesterday. And there's ongoing phase 3 clinical trials with this particular drug at 70 sites in 17 countries. And we're hoping that this will be, a drug that people will be hearing about very soon as another tool to fight COVID-19. And there's a lot of other drugs and clinical trials as well from other groups. So we're going to need not just vaccines to combat this, because this is going to be with us for a long, long time. We're also going to need drugs. Similar to influenza, you need vaccines and you need drugs. And hoping within 6 to 12 months, there will be drugs out there that can be combined with the vaccination strategy that we can use to combat this virus.
Thank you. So we now have another question. So today, PharmaMar published result of phase 1 and 2 trial. Is it necessary to make a phase 3 trial, or could it be possible to commercialize plitidepsin in US based on these results?
And this was a paper I just alluded to. Actually, Marco and I worked with PharmaMar to put this work out, and I think Marco knows more about these things than I do. But I mean, we need the phase 3 trial data in order to be implementing it more worldwide. I mean, you need this placebo. The phase 3 is where you do a control group, and phase 1 and 2 is more about toxicity. So we'll need this data, but it's happening in thousands of people around the world right now. So the hope is that we will see something positive over the next couple months, and then this can be, as I said, another drug in the arsenal to combat COVID-19.
Yeah, thank you. Marco, do you want to add something?
Well, I mean, I really can't. I can't speculate on moving things through the phase 1, 2, 3. They have to be done in the time required without jumping steps. But I think it's similar to what we've seen with vaccines, where the general public have the impression that we rushed through everything. Of course, everything was done, that the actual phases and tests were done in the proper conditions, and it's more of the bureaucratic work that often has spaces in between that were moved or shortened. But again, for these being the kind of lab that we are, that is so far from removed from the actual placing it in humans, I can't speak for it. You know, all of the work that we did was very much the very primary testing in cell culture to find the positive hits that then had to move into more complex models, right, and systems.
Yeah, and the human cell work was done with Marco and Adolfo. A lot of great mouse work was done with Adolfo in New York, and now the experts are taking it into the clinical trial world, and hopefully we'll hear something positive on that drug and many other drugs very soon.
Thanks. So I have another question from the audience for all of you. So if you were infected with COVID, would you inject Alpidine or Remdesivir? And we already know that Dr. Adolfo Garcia Sartis would do. So, and you?
I'll let Marco answer that.
Well, I mean, ultimately, the answer will be I'll follow with whatever physicians suggest. But my other answer usually is, this is half joking, but it's ultimately even with the fear of people not wanting to take the RNA vaccine. And I said, well, you know, in my youth, I've put more illicit substances in me without asking too many questions. So I'm not going to raise too much of an issue for something that's actually FDA approved.
I got nothing to add to that.
Yeah.
Okay. So we don't have any more questions from the audience. Do you want to add something before we wind it up?
Marco, do you want to start?
Well, I mean, I just have to say, at least, you know, from the world of virology, this was this was certainly an experience I don't want to see again, but it was the most invigorating year and the most difficult and tiring year. I'm looking at my image, and these bags have not left under my eyes, no matter how much I stretch my skin. But it's, you know, to see even in my own lab, the excitement and high octane adrenaline of all of the members of my group, all the young people who, you know, for this, you know, what a better time in your life to be a virologist than now, when you no longer have to explain to the person standing in line next to you what a virologist is. It's very clear, right? But it's come to the point where it's we really need a break. We need to step back and get a little bit of rest. And it's not just the virologists that worked on coronavirus, because ultimately before the pandemic, there were just a handful of coronavirologists in the world. And then every one of us, even in all of virology, there must be probably only hundreds of teams around the world with thousands of members. We all had to either decide, do we keep our lab open and running and try to work on this virus? Or, you know, can we not do that, not be very helpful, but then step back and actually run virology for while all the others are doing the other thing? What does that mean? Well, our group dropped everything we did and entirely worked on this new virus, which meant everything we did before, even for reviewing papers, writing projects for all the other viruses, are gone. So there were other groups. You know, I'm thinking of people like my idol and friend Julie Pfeiffer. She decided not specifically to work on coronavirus. Rather, she undertook the massive review process where you have ultimately hundreds and hundreds of articles now coming in, none of which can be reviewed by people like me that simply don't have the time to do it. So they're taking up the slack, you know, and so all of virology for the last year and a half have been working exhaustively on both of these things. And it's an exciting time. I think that all of the young generation now seeing this, we're going to see in about 20 years, a whole group of people pouring into virology, which is what happened to me when I was 12 and saw on TV for the first time HIV appearing. And it was the other pandemic that really caused the earth to stop and start to focus on what was a very, very scary disease at the time. So I think it'll have it'll have a good effect overall in the kinds of people that come into science in the next generations.
And I guess looking forward to, in terms of these good effects that the pandemic will cause, obviously it's been a great tragedy, but there are a number of silver linings that are becoming apparent, and Marco's touched on a few of them. One to me is that science is in the forefront right now, and the hope would be that would translate into funding. It's demoralizing to hear that Europe's decreasing funding in science. I mean, hopefully that trend gets switched around. But there are other things that we've learned as well. And I mean, for me personally, the realization that the different disciplines are actually more tightly connected than previously realized. Like the fact that structural biologists and genomicists and virologists can actually work together more seamlessly than I thought a lot of people thought. Basically, it was out of fear. It was like, let's try this. Let's take that leap of faith that Marco was talking about. And it turns out we're more connected than we previously thought. So now, as the dust settles on this pandemic and we focus on being prepared for the next pandemic or focusing on other diseases, I think 20 years from now, we'll look back and say, because of this pandemic, we're going to have so many more treatments for so many more diseases because we learned how to connect more effectively. And also going forward, there's a picture up here from a piece that was commissioned from QCRG that Jacqueline Fabius wrote about what happened with QCRG. And it was really focused on breaking down silos across these different areas. And it's a realization when you break these silos down across different labs, across different countries, between the academic world and pharmaceutical companies, you can see how fast we can move. We're talking about a virus we didn't even know about a year and a half ago. Now we're discussing drugs being put into people and vaccines being had for this virus. That's just unheard of, right? That's unprecedented. And that's because, due in large part to the collaborative effort that went into studying this virus. So the challenge now is for us is to keep this infrastructure in place and the spirit in place so that we're better prepared, as I said, for the next pandemic. And, why aren't we working on all diseases like this? Breast cancer, Alzheimer's, Parkinson's, heart disease. And I think we will. And the goal is to keep maybe not the exact pace that we've been doing the last year and a half, but maybe the same type of enthusiasm with maybe more measured breaks in between. But I'm excited about the future based on what we've learned during this pandemic and applying it to science in general going forward.
Yeah, I think we can take a French pace next time around.
How about a Canadian pace in between the United States and France? That's good.
We just need to have a little coffee break and enjoy
That's
some.
a croissant and a coffee, Marco.
Again, it's France and United States as countries and as cultures have had a love affair ever since the beginning, and so it's a natural fit for this kind of thing to happen, even in science, even more so between Paris and San Francisco, which are really to the two cities most compatible with each other within each country. So I think, if anything, we're a good model for the world to join in, right, and bring in other groups and other countries.
Yes, thank you. So I have a very good perspective ahead of us, and I'm sure we're gonna do an excellent job in the future. So thank you very much for being part of this discussion today and for sharing your experience. It was very interesting, and also thank you for all the work you did with the COVID. So I hope this chat will help some of us to foster more scientific collaboration across borders. I want to thank you as well, viewers, for joining us. And while we are here, I want to welcome you to subscribe to the QBI TV to stay tuned for the next episode. So thank you, all of you. I really appreciate this discussion, and I wish you a good evening and a good day if you are in San Francisco. Thank you very much.
Thanks, everybody.
Bye.
Bye-bye.